Let's start.
It's okay? Okay, so I'm Anne-Sophie Ay-Berthomieu. I'm working for Motif. I would like to thank Gabriel for inviting me today. It's a really nice idea. So I will just talk to you a little bit about Motif, of course. Then I will explain to you the technology of ATACseq. Also, what are the traps of this technology when you use it in the lab? I will show you two publications using the ATACseq and the kind of results you can obtain. And I will show you also what you can do next after ATACseq and how to develop your project from your first ATACseq results. So Motif has been 20 years in epigenetics. Our headquarters is in Carlsbad, California. We have different offices, one in Waterloo, Belgium, one in Shanghai in China, and one in Tokyo in Japan. And here in California, we really have everything. So all the lab for research and development, but also all the production, all the marketing team, and all the vice president, CEO, etc. Okay. So over 20 years, we try to extend the company. So first, we really focus on nuclear function, but in 2006, we acquired IP Bio, and in fact, it was a huge collection of antibodies, and in particular, antibodies against histone post-translational modification. And it's when we really start to focus on epigenetic and not only nuclear function. In 2010, we acquired Gene Pathway. So it's when we start to offer services. It's end-to-end services. You give us your samples, and we give you fully bioinformatic analysis. So all the services are also performed in California. In 2013, we acquired Switcher Genomic, so to be more in transcription regulation, but also non-coding RNA regulation, because it's a whole collection of vector, luciferase vector, either with an upstream promoter or with a 3' or 5' UTR. So you can study the activity of your promoter of interest, and you can also study the non-coding RNA, including microRNA, and their potential targets. In 2020, we start to sell a device, the Pixel. It's a multi-sample sonicator, because sonication is really important for ChIP-seq, and also for NGS in general, because you need a certain fragment length. It's a 96 well-played device, so it's really useful for high throughput assay. And now we are going also to offer a kit for RNA-seq, either from 3' or from 5'. Regarding our epigenetic services, so you can see that we have ATACseq. We have ATACseq, single-cell ATACseq, RNA-seq, and the single-cell RNA-seq. And we just now launched the multi-omic analysis. So from one sample, you can get the result for single-cell RNA-seq plus single-cell ATACseq. They are all using 10x genomic technology. Okay. We also have ModSpec, so it's to have a global view of different histone post-translational modification. It's really great when you start a project and you want to have an overview of the chromatin. We also launched IC, so it's a 3D structure of the chromatin, for those who don't know what it is. We have also services for DNA methylation, so RBS, anchor-based bisulfite sequencing, targeted bisulfite sequencing. and we soon will have the ATACseq. So the ATACseq, it's also for single cell analysis, and in fact you use antibodies to be more precise in the definition of the different subpopulation. Okay. The RIME and the ChIP-seq. So these two technologies are really comparable. You immunoprecipitate the chromatin. So for ChIP-seq, you analyze the DNA, and for RIME, you analyze the cofactor by mass spectrometry. So if you combine both technology, you have all the transcription factor DNA interaction plus all the cofactor involved in this transcriptional mechanism. So let's talk now about epigenetic. So epigenetic is a study of how your behaviors and environment can cause changes that affect the way your gene works, but without changing the DNA sequence. Okay. I think you all know that. So just few examples that are really obvious. So for example, in a beehive, all the bees has exactly the same genome, but you can see that you have 3 different phenotypes from the same genome. The same with monozygote twins. In fact, we were able to see that in homozygote twins, you have different DNA methylation patterns. So in fact, epigenetic can read the code, but also create the phrase, the sentences. So it allows your cells to understand how to read your DNA and when to read it. So basically, epigenetic includes different fields of investigation. So usually, the first step for a lot of people who don't know anything about epigenetic is DNA methylation, because it's quite easy to analyze. Then you have also histone post-translational modification, so either a global analysis or ChIP-seq, a chromatin remodeling, and the 3D structure of chromatin. So this is the 3D structure of chromatin with the IC. Now you can have a lot of data. It's quite interesting because it allows you to detect the trans-regulatory domain of your gene, whereas with a ChIP, usually you are close to the promoter and the gene. So it's another level of regulation. So let's focus now on ATACseq technology. So basically, ATACseq identify open chromatin region. You can selectively sequence transcriptionally active region, since we all agree that open chromatin region is transcriptionally active region. So why map open chromatin? So analysis of open region of the genome as a surrogate for activity. So it's also usually easier than doing ChIP-seq with a transcription factor, for example. You don't need any nuclease or sonication assay, so it's unbiased, because we know that digestion can create a bias in fragmentation, and sonication can be complicated to set up in a lab. And you have the possibility to compare healthy and disease state. And it's really easy with peak calling analysis for bioinformatics. And you don't need any antibodies. And I guess that you are all working on species that are not mouse, rat, and human. So it can be really challenging for you.
They are better than humans.
So it can be really challenging for you to find the right antibody. But with ATACseq, you don't need antibodies at all. So I will first explain to you what is the TN5 transposase. So the TN5 transposase is really an amazing enzyme because it can cut DNA and also ligate the adapter to DNA at the same time. So that's why the ATACseq technology is so easy to set up in the lab, and it's really fast. Because in one step, you do everything. And in fact, it's really elegant because it's only based on stoichiometry. So in fact, the TN5 can also access open chromatin. So there is nothing complicated in that, but at the end, it's really clever, you know. So how goes the assay? So after preparing your nuclei, the TN5 transposase gain access to open chromatin. Then it cut DNA and insert the adapters for the sequencing, okay? And then you get a library that you are going to amplify, and you can go directly to the sequencing. So it's 3 steps, and it takes more or less 4 hours. Maybe a little bit longer depending on your sample. So this is the kind of result you can obtain. So in this experiment, you have control treatment 1, 2, treatment 1 plus 2. So you can see that here you have an additional peak with the treatment 2 that you cannot observe with treatment 1 or control, suggesting that you have a new open chromatin region with this treatment. And here it's also interesting because it's partially closed with the treatment 2 as compared to treatment 1 and control. So I also talked to you a little bit about ChIP-seq. So what are the advantages of ATACseq versus HistoNAP ChIP-seq? So first is the cell number. Usually with our kit, or even in general, we recommend to people to use at least 1.5 million cells to start with ChIP-seq. For histone modification, you can go lower, but if you want to be in the best condition, it's better. With ATACseq, you can use only 50,000, 100,000 cells, so, and even 1,000 cells, apparently. So you don't need any antibody, and the time also is shorter, since with ATACseq it's 4 hours, and for ChIP-seq it's 2 days, because you need to incubate the antibody overnight. And also the cost is lower because of the antibody, because the sequencing is deeper, the quantity of samples is higher, so if you treat them, you need more treatment. So overall, ATACseq is really less expensive and really cost effective, in fact. So what are the traps? So the first really important step is the sample preparation. So first, the quantity. So we recommend 5,000, 100,000 cells and between 20 and 50 milligrams of tissue. For cells, you can go a little bit lower. For services, we were able to accept 10,000 cells, depending also on how it's easy to light the cells or the tissue. The nuclei isolation is really important. If you have a lot of cell deaths, apoptosis in fact release free DNA in your samples, and in fact the tagmentase is going to cut this free DNA, and you will have a lot of background. So if, for example, you are using a treatment that leads to cell death, you can use DNA before isolated your nuclei. Okay, so you remove all the cell-free DNA that you have. The other issue can be mitochondrial DNA, because it's going to have a lot of reads. For mitochondrial DNA, you need to treat with digitoline. It will paralyze the cells and allow a better nuclei isolation. Then the tagmentation reaction. An easy way to check the tagmentation reaction, it's a library QC. So, and also the quantities that you are supposed to have. So if everything goes perfectly in your experiment, you are supposed to obtain between 400 and 600 nanograms of library, okay, after amplification. Regarding the fragmentation distribution, you need to have a smear between 200 and 1,000, sorry, forget a 0, 1000 base pair. So this is the perfect QC, okay? And you need to have the majority of your fragments between 200 and 600, okay? So for example, if it's under tagmented, you can see here, in fact, you have not really a smear decreasing with the increase of the fragment size. Everything is between 2,500 and 250. So this is not good. So when you get that, you can also improve by purifying smaller fragments of your library, but you will lose quantity. So let's talk now about some publication. So this one is one of the, it's the first publication of Wenrostro and Greenleaf, and it's also from this paper that we develop our own technology at Active Motif. The goal of the ATACseq was really to enable real-time personal epigenomic, which is really a dream, I think. Imagine you treat a patient and every day you are able to follow how the treatment is going. So this is really great. So here they just have to get some blood, and after ATACseq, look at the sequencing. So here is just one of the results. I did not explain all the paper, of course. So during this daily monitoring, in this case, they see that the promoter for interleukin-2, a cytokine that drives T cell growth and inflammation, is monitored. So you can see 1, 2, 3. So you can see the peak of open chromatin are increasing. And here you can see that NF-AT binding park of the EL2 locus is open, so suggesting that NF-AT can bind to this locus. It was further confirmed with the ChIP-seq in Jurkat after cyclosporine A treatment, and they can't see anything regarding IRF4 and STAT3 transcription factor, and you can see that with the ChIP-seq, they didn't obtain anything. So it's also a good idea when you are performing ATACseq to then focus on a specific locus and perform ChIP-qPCR instead of ChIP-seq. It can be also increase your knowledge about all the molecular mechanisms. So I also choose another paper regarding bovine respiratory syncytial virus. So here they use this scarf branchial lymph node, treated or not, challenges or not, with the BRSV, the virus. They analyze these samples with ATACseq and compare the data with an old publication using exactly the same protocol, and they get some RNA-seq data. So this is really something classical also we have as a services platform. It's ATACseq plus RNA-seq and data integration of both experiments. So first, they observed that 50% of the region of open chromatin were located less than 2 kb upstream protein coding genes. So it sees the confirmation that open chromatin is transcription active region. They also find that more than 8,000 regions are open only in the BRSV challenge lymph node. And in this chromatin region, there is 174 genes in this region that are also differentially expressed in the RNA-seq of the previous study. So they choose this gene to perform ingenuity pathway analysis, and they get all this enriched biological process. So you can see that most of these processes are involved in immunological response, which is not really surprising since it's an infection. So, yeah, I will not go deeper into bioinformatics because I'm really a wet lab person and I don't really understand anything about this. But you have a lot of software that are able to give you this kind of graphic at the end. I wanted also, I just did one slide about that, but another alternative to ATACseq, it's single-cell ATACseq. So this is the standard genomic workflow. So it's really nice because you have barcoded bits. Here you have your nuclei where you perform tagmentation. Then you mix everything, and each nuclei will be tagmented with a specific index. And then you go to sequencing, and you are able to analyze the sequencing data of each cell individually. So when using that, so when you have heterogeneous tissue, for example, typically the liver contains a lot of different cells, for example, so it can be interesting to perform this kind of analysis. It's also avoid any cell sorting, which is long, fastidious, and also induce cell death. It can be also interesting in dynamic processes, for example, wood healing, development, things like that, because you can really, in one organ, when you have different cellular at different states, you can identify them. And it's really powerful because it can detect the subpopulation representing only 2% of the total population. So in summary, open chromatin is active chromatin, and ATACseq can map this on genome-wide level. ATACseq data provide distinct peaks, and you can see it's really easy to compare one treatment to another. It's also a really easy assay, really short in time, and really cost-effective. And finally, if you have a good bioinformatician, you can detect active transcription factor binding site within promoter and informing you of the transcription dynamic. So what to do next? So after ATACseq, as I told you before, you can perform RNA-seq. It's really complementary. You can also go for cotton tag and chip-seq. So cotton tag is another TN5-based technology. So it's to replace chip. So the TN5 is in fusion with the protein A, which is able to bind to antibodies. Okay. So in fact, you immobilize your cells. You come with your primary antibody, secondary antibody, the PRTN5, and then the PRTN5 cut in the vicinity of the primary antibody and insert the index for sequencing. So it's also much faster than the chip-seq, and you need also less material at the beginning, less antibody, and less sequencing. So overall, it's more cost-effective than chip-seq. But for now, it really works well for histone modification or really highly expressed transcription factor or nuclear protein like CTCF, coenzyme, things like that. And of course, very important bioinformatic analysis. So I don't know how you do it in your different lab. But if you have a good bioinformatician, you are able to identify promoter enhancer, transcription factor motif, nucleosome positioning, and nucleosome depleted region. I wanted also to let you know that, so yes, we have products and services, you already know, but we also have a lot of resources with completely free access. So we have a complete guide to ATACseq. We have 2 webinars about ATACseq and single-cell ATACseq and its application. These webinars, they last like 20 minutes, I think, so it's really easy listen to. And we have also a podcast. So here is Buon Mostro, who was able to give us an interview. So in general, it's quite nice because they talk about themselves, but also how they wanted to, how they had the idea to develop ATACseq, things like that. So it's really nice to listen. So I really invite you to go to our website to see the resources. So if you have any question, don't hesitate. Thank you very much for your attention.
Thanks a lot.
Yeah.
So I'm just trying to understand the ATACseq.
Maybe a little bit louder.
Oh, So a very basic question, like I want to understand, so the attack sequencing, it sequences all open chromatin,
Yes.
or only the regions that harbors the promoters and the enhancers? Like I want to distinguish between these 2 points.
No, it's really all the open chromatin region.
And
It's
then,
not only a region close to promoters. You can see the last study I showed you, only 50% of the open chromatin region were close to a protein coding gene.
okay, and then essentially with the help of bioinformatic tools, we identify the promoters and enhancers.
Exactly, yes.
Okay.
Integrating with other different kinds of data. So with ChipSeq is really essential if you want to see if it's promoter, enhancer. You integrate the different levels, as Krista was showing in the beginning. As much as data you have, it's possible to refine and have a clue what is happening in this region.
Yeah. And also the sequencing technology essentially is a similar, just like the other sequencing techniques.
Yes, because it's exactly the same adapters and the same index. Yeah. It's Illumina, if you were wondering.
There's no question in the chat box.